obviously regions with high detergent content, and that makes the membrane break apart at these defect regions. So this is a phase diagram for a lipid and another detergent. And here you see, if you look at this thing at whatever, 45 degrees centigrade, you get more than 40% more of this detergent into membranes without destroying them. But if you do the same in the gel phase of the lipid, you will start forming micelles already a little bit beyond 10%, which also means that if you have an intermediate composition, something in this case, for this example, say 30%, then you will have just vesicles that can be nice and tight in the fluid phase. And if you cool them down, you end up in the mixed micellar region. That means you would open up, you would destroy your liposomes above the trigger temperature, below the trigger temperature. So this is just the opposite of what we just looked at, what was the idea how lysolipid would act in thermoresponsive liposomes. So this is one aspect of what we call the Lysolipid paradox. So the question is, why does lysolipid act like a detergent in the fluid phase, but fundamentally differently in the gel phase? And how is it tamed? How is it made not destructive upon storage and at body temperature? So again, I mean, it is hard to imagine that something that fits so badly. I mean, in the gel phase, all the tails are stretched out and packed together. And it's hard to imagine to have 10% of lysolipid mixed ideally with a gel phase. So this is kind of not really convincing. And the question is, what's going on? There is another paradoxical thing about the lysolipids, and this is the release characteristic. So if you have your liposomes loaded with a dye or with a drug, and you have them at room temperature, and now you bring them to a certain temperature, and you measure the release as a function of time, you get release curves like this. So you get a reasonably fast release in the beginning, but then the released material is reaching a constant value less than 100% potentially. So that means the membrane is opening up, you get pores that release the drug, but then, and you're still at trigger temperature, the membrane anneals again. So the question is, how would this work with the detergent-like pores you just have seen in this attempt of an explanation? So what makes the membrane anneal after a little while? That's the other question. So we looked at these systems carefully just by studying phase transitions in these different mixtures, and I don't want to go into too much detail here. This is just differential scanning calorimetry and what they call pressure perturbation calorimetry. Doesn't really matter. The important thing is we see transitions. We see the transition of the pure DPPC here. Then if we add 5% lysolipid, we see a broader transition that is occurring at lower temperature. This is fairly normal. But the surprising thing is if we go from 5 to 10% in lysolipid, this effect is not continuing, but we get starting at the same position, but we get a narrow peak again. So we have a composition where we have a narrow peak again, and then if we add even more lysolipid, we get peaks that start at the same point but have these enormously broad shoulder up to almost 45 degrees centigrade. If you would have a system with 50% lysolipid, you get a sharp peak at these 46 degrees centigrade. So now we just had our final exams in pharmaceutics in Freiburg a couple of weeks ago, and one question in the exam could have been, what specific mixture shows a sharp melting point below the melting points of the individual components? And since you probably all passed your exam at some point in the past, you of course know the answer to this question. This is a eutectic. So obviously we have a eutectic right at the composition that has evolved to be the best composition for the thermoresponsive liposomes, 10% So this is the lysolipid that stands for monostearyl phosphatidylcholine. So just at the 10% that is being used for the drug delivery system and 41 degrees centigrade, which is the trigger temperature in the body, we have a eutectic point. So this is why we get this narrow transition. And at this eutectic or below this eutectic temperature, we obviously have a coexistence of two phases. One is a gel phase bilayer that contains of the order of 5% lysolipid only. And the other one is a phase that contains almost 50-50. Under these conditions, it's more 30% or 33%. So we have another phase that has an enormously high lysolipid content. And in fact, it is not so much of a puzzle what this phase is, because it has been shown already a long time ago that if you have a 50-50 mixture, you get these interdigitated phases. Funnily, actually, David Needham was one of the co-authors of this paper, so the person who was also taking a leading role in developing this drug delivery system. So the idea is the following. A head group needs about 65 angstrom squared in area. A stretched chain needs of the order of 21 angstrom squared. So one head group can nicely cover the area of 3 chains. So if we have them, if you have a monolayer and some head groups on this side and some head groups on this side, and you can see that you can do this best at a 1-to-1 mixing ratio, then you can get a very stable phase. You don't even need to tilt the chains. You get something very stable with an enormously high lysolipid content, but which is very stable, not leaky, everything fine. So this is more or less the idea what's going on. And of course, we checked the X-rays again at 50-50 content. We found pretty much what has been shown for a different lysolipid, C16, in this McIntosh paper 95. So we see an interdigitated phase where actually the thickness of the membrane is considerably smaller, and you don't have any methyl region in the middle, as compared to the blue one, which is for the DPPC, where you have a thicker membrane with this dip in the middle in electron density. But the crucial thing is now, if you look at this with 10% lysolipid, we see a superposition of the peaks of the reflexes we have at we have for the interdigitated phase and we have for the bilayer gel. So at 10%, we see a coexistence of interdigitated domains in a bilayer of gel phase. So what's going on is the following. At storage temperature, you have these domains. Everything is safe. You have only a very small content of lysolipid in the gel bilayer. At 41 degrees centigrade, now everything is melting at once. That's the idea of a eutectic. So you get an enormously high lysolipid content in some spots. There you are forming these pores that are releasing the dye. But if you wait a little longer, of course, the lysolipid can distribute freely over the membrane. And this is the idea what makes the membrane anneal. If you just spread out 10% of lysolipid evenly in a fluid membrane, it is not causing pores anymore. So this is what makes the membrane anneal. So this is my conclusion. This is actually what's going on. This is explaining why you can have stable, tight liposomes below 41 degrees, and it's explaining why you have this incomplete release. You can think about what would happen if this liposome has passed the hypothermal region, goes through the body, and an hour later comes back to the hypothermal tissue. I want to thank Phospholipid Research Center for support of this project, and I want to mainly, to have seen there are many authors, I want to mainly mention Daniel Eckert, who was the one who did most of the experiments with the DSC and PPC and started the whole thing as part of his doctoral thesis. Thank you.
Thank you, Heiko, for this nice talk about the Lysolipid paradox. And since we have a couple of minutes time, are there any questions about the Lysolipid paradox? Maybe I can start with the first question. Or is there any question? Yeah, in the back. Please use the mic. Please use the mic because we also stream this session online and the other one cannot hear you without the mic. Thanks.
Which will be the behavior in a bilayer that it's a room temperature fluid? Would you expect then directly this distribution homogeneously?
So you're talking about POPC or another lipid that is in POPC, you would tolerate something like 10% of lysolipid in a bilayer without creating much trouble. Lysolipid has another strange feature, and this is that it's not doing flip-flop across the membrane. So if you add it on one side, you get into other kinds of trouble with bending membranes. But when you have it in both leaflets, you should tolerate 10% in a fluid membrane without causing too much trouble.
I have a second question. So my question is this effect also evident when we use a higher TG lipid like DSPC?
I mean, if you use DS, I think you will have the same principal behavior. Then the transition temperature is shifted a little up in the temperature range, which is not essentially what you want because the 41 degrees is perfect for body temperature to hypothermal tissue. But the principal behavior would, well, we have not studied this in much detail, but it should be similar.
Okay, thank you.
Yeah, thank you. What would happen, because I worked with these liposomes quite a lot with thermodox in vivo and after intravenous injection, and of course then you have a lot of protein binding. Do you have any idea how this would be affected by protein binding? Because we see also incomplete release and we see actually spontaneous release, but incomplete without going up in the temperature. So it's not in the high full exposed area, it's mostly the tumor, but already spontaneously after injections, there's quite some release.
Yes, this would be good for another couple hours of talking. It is. I mean, this is explaining what's going on in the vial.
This is also, I think, very good that we know the mechanism, but
I mean,
it's
if you
more
have it in the
complicated
bloodstream,
if you use them.
If you have it in the bloodstream, you will have two effects. One is that albumins, for instance, are very good at binding lysolipids. So they will extract lysolipid from the system. So you will have less lysolipid in your liposomes. And the other effect is that albumins are binding to the liposome surface. And this may, for instance, contribute to keeping such pores open that would close otherwise. So the albumin interaction can actually also boost the release further than what you have in vitro just with the lysolipid by different mechanisms. Yes, if you really want to understand the release curve in blood, you have to definitely look into the albumin. And we did a little bit in this direction as well. You have to look into the albumin.
Yes, sure. Okay, last quick question.
Thank you for your interesting talk. Do you think that, because usually we would add some pegylated lipids, do you think that this would also influence the percentage of lysolipids that you need to add into a system to keep this from happening? Because it would prevent a lot of albumin binding.
Yes. You have also in this LTSL mixture, you have a little bit of a pegylated lipid to stabilize your liposomes. And again, this is having some qualitative effect or some quantitative effect, if you wish. So it shifts the phase transition a tiny little bit. And it also affects the opening time of these pores because, I mean, the pegylated lipids themselves are also micelle formers, so they will also like these. But again, we are convinced that it's not changing the principal behavior. So I think it is not going to question the existence of these interdigitated domains, but it's going to be shifting things by half a degree or things like that.